All right, so I want to start by just kind of refreshing what we talked about yesterday. So yesterday we talked all about the proteins, that proteins do everything basically. So your DNA has to code for proteins. And we introduced the central dogma of biology yesterday that just is DNA codes for RNA, which codes for proteins. Okay. And does that sound familiar? Right, like the big idea of yesterday, DNA codes for RNA, which codes for proteins. So then, at the end of class yesterday, we did that coding activity together, where we were getting comfortable reading codes, and we're going to apply some of those rules to reading your genetic code. So today, that's where we're going to pick up. Okay. So after we figured out the pattern of information flow in a cell, meaning this pattern, how we go from DNA to RNA to proteins. The next step is to figure out how exactly do the sequence of bases in mRNA code for amino acids that make up a protein. And so like the code for RNA is just going to be those base pairs, so like A, U, C, and G. So how does your body read that and somehow figure out amino acids from there? Okay. So the genetic code has to contain rules that specify the relationship between a sequence of nucleotide bases in DNA or RNA, and then the corresponding sequence of amino acids in protein. Okay, so we're going to look at what are those rules to read our genetic code. So the first thing we have to figure out is how long is a word in our genetic code? So remember yesterday we had to figure out like how did you distinguish one word from the other word? So your body has to do the same thing. So George Gamow predicted that each word in our genetic code is going to contain 3 bases. And he did this just by using some math. And I'll show you on the next page, like, how did he figure out it was just 3 bases. But then the short story of how he figured out it's 3 bases, we know that there are 20 amino acids. And so the smallest number of bases that could at least make up 20 different codes had to be 3 letters. Okay. And so his math is there's 4 letters, A, U, C, and G. Okay. And so if you have 3 letters in like a code sequence, 4 times 4 times 4 equals 64. Okay. So 64 options is enough to code for 20 proteins. And so since this code consists of 3 bases, it's just called a triplet code. So triplet means 3. So here's just a diagram to walk you through his thought process. Okay, so we know there's 4 bases in RNA. So A, U, C, and G. If there was just one base equals an amino acid, then you just have 4 options. 4 options is not enough to code for 20 amino acids. The next option, what if it was just 2 letters? 2 letters coded for an amino acid. Well, again, we still have 4 bases. So 4 times 4 is 16. If you just did 2 letters per base, you only get 16 codes, but there's 20 amino acids, so it has to be more than 2 bases in a code. So then he moved on to 3 bases. So if you have 3 different bases in a code, you just take 4 times 4 times 4, which gives you 64. And then 64 is finally that lowest number that's finally greater than 20. So then he proposed it has to be 3 bases in a code that could code for all 20 amino acids. So then a triplet code, it has to be redundant, and I'll show you what I mean by that in a couple slides. Okay. But all it means is some amino acids can be identified by more than one triplet code. And so 3 bases make up a word in our genetic code, but instead of calling it a word, we call it a codon. Okay. So words are now codons. Okay, so your body is going to read codons. So now we have to move on and figure out, well, what's next? How do we still read this? I know I'm reading them 3 at a time, but I also have to know when to start. Okay, so remember yesterday, our codes all started with the word run. That was like your indicator that it's time to start. Well, we have learned that there's a special start codon. The RNA sequence is AUG. Okay, so whenever your body reads a strand of RNA that says AUG, it tells your body to start making proteins. And then in addition to knowing when it's time to start making proteins, it's equally important to know when it's time to stop making proteins. So there are 3 stop codons. Okay, so UGA, UAA, and then UAG. And so once your body reads any of these 3 codons, it's telling it to stop making protein. Okay. Or kind of like yesterday, what was the word at the end of those codes that told you it was done? Done. Okay. So then these are the codes that your body reads. So if we go back to our options, 4 times 4 times 4 is 64. So there's 1 that codes for start, 3 that code for stop. So that means that there's 60 options left. So then those other 60 codons are going to make up the 20 amino acids. So here's a chart of how to read the amino acids. I'm going to hand this out to you, and then we'll go through it of how to read it. Okay, so I want to go through this chart, and I want to show you how do you read the codons to figure out what amino acid they code for. You do not have to memorize any of this. Okay, so you don't have to memorize the codons. You don't have to memorize the amino acids. You will always be given a chart, so you just need to know how to use the chart. Okay, so this pink copy I just gave you, this is yours to keep in class. So like when we do problems in class, you're going to need this chart. When it comes time to take a test, I will give you a chart or I'll like print it on the test. Okay, so no memorizing, you just need to know how to use it. So if you look at your chart, this column is for the first letter. This is for the second letter, and then this is for the third letter. So let me show you an example. Let's say we want to look at this codon. Let's say it's CUU. So I go to my first letter, that's C. Then I line it up in the column for the second letter. And then I line it up in the row for that fourth letter. Okay. So C, U, and U all line up right here. Okay, so do you see where I get CUU? And then it codes for this amino acid. It's called leucine, or you just get the 3-letter abbreviation LEU. So now let's try to find one. Let's say it's AAA. So use your chart. Let's try to find where's AAA. So we start with the first letter A. I'm going to go over until I line up in the column for A. And then I'm just going to go down until I find the third row for A. Okay, so AAA is here. It codes for the amino acid LYS, which is lysine. Let's do one more. Find GGA. What 3 letters code for GGA?
GLY.
Good. Okay, so we go G to A, which is GLY for glycine. Okay. Do you think you could like use this? You can find amino acids. All right, so then as we looked at that amino acid chart, there's a few properties that we need to know about for a genetic code. So first of all, it's redundant. Okay, so that means all of the amino acids except for 2, they can be coded by more than 1 codon. So what I mean by that, if you look at your chart, just look where like UUU is. If you see UUU, that's for SER or serine. Do you see how there's 4 different codons that code for serine? Okay. So that's what I mean by it's redundant. It's unambiguous, which means one codon always codes for the exact same amino acid every single time. It's never going to code for one amino acid in this case and then a second in a different case. It's always going to be the exact same amino acid for the exact same codon. It's nearly universal, which means the codons that code for amino acids in my body are going to code for the exact same amino acids in my dog. Okay. There's very few exceptions to that rule. Okay. So that means, again, if we go back to UUU, it's going to code for serine. That's going to code for me as a human, in my dog, in my cat, and all of it. Okay. And then finally, it's conservative. So when the same codons code for the same amino acid, they usually have the same first two letters. So again, just go back to the same box we've been looking at. I've been looking at the UCU box. Let's look at UCU. That's the one for serine. If you look at that box for UCU, do you see how all of them start with UUU and then CCC? Mm-hmm. Okay, so that's what we mean by it's conservative. The first two letters are the same. So now that we know how to read our genetic code, now we're going to really get into transcription and translation. So remember, it's this process of how do we get from our DNA to our mRNA and then mRNA to proteins. So now we're going to look at the actual details today. So just to remind you, when a cell builds the proteins it needs, the instructions come from the DNA, and this is all according to that central dogma. My words got cut out, but I don't think you need them for your notes. Correct? There's just central dogma. And this picture should look familiar, right? This is our DNA. Through the process of transcription, we get mRNA, and then through the process of translation, we get a protein. So then transcription is just that first step of converting the information stored in DNA and being able to convert it into a strand of mRNA. And the enzyme responsible for synthesizing that RNA is called RNA polymerase. And the strand that RNA polymerase makes is called the template strand. So think of it, it's like a template that's going to be read in the next step of translation. All right, so here's a picture of DNA being converted into RNA. So this red strand at the bottom is your DNA. This yellow strand is the RNA. Before we read this, I just want to refresh. What are the 4 letters of the base pairs in DNA? Okay, we have A, T, C, and G. What are the 4 letters in RNA? Okay, we have A, U, C, and G. So I'm going to show you how that difference is going to play a role here. So when we look at our DNA, here are the base pairs of DNA. So we have G, G, A, T, C, and C. So when RNA polymerase makes a new strand of RNA, it just attaches the complementary pair. Okay, so if DNA has a G, what letter is complementary to G? C. Okay, so the RNA is going to have C, or a cytosine. Here in DNA we have G, so what letter is complementary to G? C. Next, we have adenine or A. What letter is typically, if it was DNA, what letter would be complementary? T, but there is no T in RNA. Instead, it's U for uracil. So A is now going to pair with U. Here we have a T. What letter is complementary to T? Okay, so when A would go here. What letter is complementary to C? G. And what letter is complementary to C again? Okay, so RNA just polymerase comes in and it puts these new base pairs there. Just like DNA polymerase, RNA polymerase also moves in the 5' to 3' direction. And then I've added a slide for practice. Okay, so this isn't on your notes, but find some empty space. You can find space on this note packet. You could use the chart I just gave you. I want to practice converting this strand of DNA into RNA. Before we do that, I just want to write the base pairs down. So what base pair of DNA is going to be complementary to RNA? I feel like this could be helpful since we're just changing it up with a U in there. Okay. So what are the 4 base pairs of DNA? Okay, AT, G, C. Okay. So the complementary RNA base for A would be what? Okay, so U. And then what's complementary to T? A. What about G? C and G. Okay, So it's not a bad idea to write this out on your paper, especially if like the U is throwing you off. Okay, so you can always write this out. So now I just want to go through and I want to write the RNA strand. So if DNA has a letter A, what's the complementary letter for A? Okay, so I'm going to have a U. What's complementary to T? Okay. What about C? G. What about G? C. What comes next? The next letter is G, so what's complementary? Okay, what next? Okay, U. Then what? Wait, what's complementary to C? G. Then what's complementary to T? We get another A. What's complementary to A? So U. We get a U, C, G, A. Okay, do you think you could do some of these on your own? Okay, this is transcription. So when we talk about transcription, we're just talking how RNA polymerase comes in, it reads a strand of DNA, and it produces a complementary strand of RNA. Record this. Okay, I took a few slides out. So take a look at your note packet. Jump down to where it says an introduction to translation. And we're not going to do this. I just tried to simplify the notes a little bit and eliminate some extra info. Okay. So does everyone see on your packet where it says intro to translation? Yeah. Okay. So now translation is that second step. It's where we take the information in mRNA and convert it into that amino acid sequence to build a protein. And all this happens in the ribosomes. So that's where we translate mRNA and actually build the protein. So here's just a picture of a cell again. Okay, so inside the nucleus here, this is where we have our DNA. So RNA polymerase comes in and it reads the DNA and it makes that complementary strand of mRNA. mRNA is then mobile, so we can move out of the nucleus into the cytoplasm where it meets up with the ribosome. And then the ribosome is going to read through and translate the mRNA and build a protein. Okay. So now we're going to shift gears and we're going to look at these stages of translation. So we know that during translation, something is going to have to read the mRNA strand. And we know that 3 of those base pairs make a codon. And we now know that those codons are going to equal some sort of amino acid. That's what we just did on the chart. So Francis Crick proposed that there has to be some sort of adapter molecule here that can actually do this process. It can read the codon and match up the amino acid. And so it turns out subsequent work showed that there is indeed an adapter molecule, and that adapter molecule is called transfer RNA or tRNA. So tRNA molecules, we say that they're charged with an amino acid on one end. So think like towards the top, it's going to carry an amino acid up here. And at the bottom of the tRNA, it's going to have this thing called an anticodon. So it's going to have 3 letters, and it's going to be complementary to the codon it needs to read an mRNA. Okay. So by complementary, if the mRNA is the letter U, the anticodon is going to be the letter A. If the codon has C, the anticodon is going to be G. And if it's the letter A, the anticodon will be U. Okay. Both of these are RNAs. So again, there's no T's. It's only U. Okay. And so because the tRNA has this anticodon, this is how it's going to be able to figure out which amino acid goes where in the sequence. And then these next few slides, we're going to go more in depth with this. Okay. So all of this happens inside the ribosome. So I just want to talk about the structure and the function of the ribosome real quick. So ribosomes are made up of just proteins and ribosomal RNA, which we just call rRNA for short. And so over here, this is a picture of a ribosome. It's like that red-brown color. You can kind of think of this as like a big, poofy hamburger bun. So there's like the top. That would be like the top of the bun. then there's the bottom. And so the mRNA goes in between the bun. Kind of think like where your burger patty goes. And then inside the ribosome, I know it's a little tough to see, but do you see how there's 3 slots here? Do you see how there's three little indents? So that's a site for 3 different tRNA molecules to line up, and they're going to participate in helping make a protein. So those 3 sites, they have names. So the first site is called the A site, and A just stands for acceptor. Okay, so this is where a tRNA molecule is going to be accepted. Okay, kind of think maybe it enters here. The middle site is called the P site. This is where a peptide bond forms. It's when you put all those amino acids together. We call that a polypeptide chain. By the time it's finished, we'd call it a protein. And then the third site is called the E site. This is after the tRNA has dropped off the amino acid and it's going to exit. So here's just like a blown up picture of a single ribosome. So here are the sites, so A, P, E. Read it from right to left. You enter on the right, you work your way to the left. I remembered it because it spells ape. Okay, so I don't know if that helps you remember it, ape. So APE. So A is the acceptor site, P is where the peptide chain forms, and E is exit. Okay. And we're going to go a little bit more in depth on these next slides of what happens there. So in that A site, okay, the amino acid tRNA is just think tRNA. Okay. So it carries the correct anticodon to match up the mRNA, and it's also carrying the amino acid. Okay, so that enters in on the A site. At the P site, a polypeptide bond is going to form between one of the amino acids and the second one. Okay, so we're going to start growing our chain. And then the ribosome is going to keep moving along the mRNA strand until you have all of the tRNAs, they just shift down one position. And then the tRNA in the furthest left spot, it's going to get booted out of the ribosome because it did its job and now it's done. I do have a video. I'm going to wait and show you this video at the end just because my screen sharing and recording software doesn't let me play a YouTube video. So there's 3 phases of translation. There's initiation, elongation, and termination. All right, the first stage is called initiation. What does initiation mean? Start, okay? So this is where making proteins would start. The mRNA has to contain the start codon, AUG. So when the ribosome comes along to read that mRNA, it sees AUG, and we know it's time to start making proteins. The second stage is called elongation. What does it mean if something elongates? Gets longer. Okay. This is the part where we build that long chain of amino acids that make the protein. So this is where the tRNA is going to enter the P site. Okay. The E and the A sites are going to be empty for the moment. Okay. Then the tRNA, it's going to bind in the A site where the anticodon is going to meet the codon. Okay, and I'll show you this in a little bit. Okay. Then another tRNA is going to move in to the A site, okay, and a bond is going to form between the amino acid in the A site and the P site. Okay, so we're going to start making this chain. So here it is just in picture form. So I'm going to start on the left here, and then I'll work my way this way. So you have your tRNA. They match up here. So this is how we know which tRNA goes where. The anticodon is complementary to the codon. And the tRNA carries the amino acid. Okay. So then a new tRNA is going to come in. Okay, this knows to come in because the anticodon here is complementary to this codon. Once it arrives here, a peptide bond is going to form between these 2 amino acids. And so do you see how my chain's now getting longer? And then next, the ribosome moves over. Okay, so it leaves the A site empty. And the original tRNA that was here, it now gets booted. Okay, so it dropped off its amino acid. It did its job. It moves. And then now a new tRNA can come in, drop off its amino acid, and this keeps going. These can be hundreds to thousands of amino acids long. Okay, so this is a long process. You build that long chain. So when the ribosome moves down the mRNA strand, we just call that translocation. Okay, so just think we're changing location. So when this happens, that A site becomes empty. So a new tRNA can move in. And so when that new tRNA moves into the A site, just a new bond forms between the P and the A. And whatever is in the E, it gets the boot. It finished its job, so it's done. Okay, so kind of think of like an assembly line, right? A tRNA, think they move up, they drop off their amino acid, they move out. One moves in, drops off the amino acid, moves out. And then finally, that third stage is called termination. What does termination mean? Stop, okay? So when the A site encounters a stop codon, one of those three stop codons, it's going to release something called a release factor. It's going to look like a tRNA. It fits kind of the shape of a tRNA, but it doesn't have an amino acid. So then this tells this sequence that it's done. There's no more amino acids to put on. So your polypeptide chain is complete, or your protein is now complete. So here it is just in picture form. So this is your release factor. So it looks similar to a tRNA. But if you notice up top here, it doesn't have an amino acid up here anymore. Instead, it's just going to break this bond here. So your polypeptide chain is complete. Your protein is finished. All of these are going to get the boot, and they're just going to end up disintegrating in the ribosome. It breaks up, think like you open the hamburger bun, and it can move on to a new strand of mRNA. So then, once your protein is released, it has to fold up, so it folds in some sort of three-dimensional shape. That structure is going to determine the function of that protein. Is it for movement? Is it for support? Is it an antibody? Is it a hormone? And then the protein, it gets sent to the Golgi apparatus, where it gets processed and shipped to whatever part of the cell it's needed. And so that's going to depend on what's the function of the protein. And then the function of those proteins, those are the traits that we talked about on our unit of like Mendelian genetics, right? That protein is going to say, does the mouse have brown fur or white fur, is what we talked about yesterday. Okay,